Hey there, my name is Todd Pierson, and I'm an assistant professor at Kennesaw State University. I'm excited to be here today as part of this session to talk to you all about environmental DNA tools for mapping Appalachian salamander communities. And before I get started, I'd just like to thank all the folks listed here in this slide who have contributed meaningfully to the various projects that I'll touch upon today. That includes Kathy Jakowski, who's a co-author of this presentation. So many of you probably already know that the moist, cool forest and the pristine, bubbling brooks of southern Appalachia are home to a tremendous diversity of amphibians, specifically salamanders. Unlike many other groups of vertebrates, salamanders are not most diverse in tropical latitudes. They're most diverse in temperate areas. And that's really driven, that pattern is really driven by the diversity of a single family, the plethodonid or lungless salamanders. And that's largely in the southern Appalachian Mountains, as you can see in this map here. And this group is the focus of research in my lab. We study the ecology, evolution, and conservation of these salamanders primarily. And I'm going to talk to you today about some environmental DNA tools that we use in the course of our research. Now, some of these salamanders are beautiful, breathtaking, and unmistakable. You will have no problem identifying them, such as this two-line salamander on the left, or the spring salamander, a red salamander pictured to its right. Even though these are easy to identify and differentiate from other species, they pose their own challenges in that some of these species are behaviorally cryptic. You can spend all day in a stream with a healthy population of spring salamanders and not find one. So that makes it challenging to study their ecology or evolution or whatever you're interested in, or plan for their conservation and management. Another group of lungless salamanders pose a larger challenge, which is that even though you might be able to find some of them, they all look the same. This is the aptly named dusky salamanders in the genus Desmonethus. And pictured here are three ecologically and evolutionarily distinct species that can, especially for novices, be almost impossible to differentiate. And even for amphibian experts can be very challenging. And so even if you can find these salamanders in the field, you often have a hard time knowing exactly which one you've got. And this problem exists even for the named species, and there's a lot of undescribed diversity in this group too. So they pose a variety of challenges for studying. And if that's true for the adults, it's doubly true for their aquatic larvae. Pictured here, the larvae of those same species. And these are, I don't know, 1,000 times life size. These are very tiny salamanders, very difficult to differentiate in the field, even for experts. And by now you've heard a little bit about the generalities of how environmental DNA works and some of its applications. I won't go over those in great detail, but I'll just show you some quick pictures of how we collect these data. So here might be one of our study sites. This is a beautiful stream in Great Smoky Mountains National Park. And we might go into the field and collect bottles of water. Ideally, you don't leave them sitting out in the sun like I did for this photo op here, as it degrades the DNA. Bring them back to the lab, we filter them, and we can later extract DNA from that filter paper and do PCR. And then depending upon the exact type of environmental DNA assay we're using, there will often be a downstream bioinformatic component to figure out what exactly was there. And so today I'm just going to highlight three applications that we've been working on of environmental DNA. And what unites all these is they're about Appalachian salamanders, but also their focus on understanding just kind of the distribution of some of these species with some downstream applications potentially later. All right. So first and nearest and dearest to my heart is the Patchnosed salamander. Perhaps you've heard of this species. It was only discovered in 2009 and described in 2013. It diverged from its closest extant relatives more than 40 million years ago. It's a very evolutionarily distinct species. It was named with its own genus. And as this photo suggests, it is tiny, and it has a tiny home range too. It's currently known for only about 25 square kilometers in Northeast Georgia and neighboring South Carolina, up near Toccoa, Georgia. And I started working on this species as an undergraduate, and we were interested in better mapping its distribution and understanding the factors driving where it lives. And so we use a traditional technique of leaf litter bag surveys. We deploy these in the streams, and we come back and check them. And we had a problem, which is that even though this method worked great for other relatively closely related species, it was not great for the Patchnosed salamander. Our detection probabilities were very low, such that we had to revisit a stream something like 60 or 70 times and check 25 leaf litter bags before we could have a cumulative detection probability of 95%. In the process of confirming that the species does not occur at a stream, we'd have to visit a lot, cause some damage to the stream as we tromp up and down, and just spend a lot of time. And around the same time, environmental DNA methods were becoming popular, and we decided it was a good candidate for these methods. And so we developed a species-specific quantitative PCR method to attempt to detect this species. And we collected samples, much like how I showed you before, with one exception or one difference. And that's that these live in really tiny streams. You're lucky if they're a meter wide and just a trickle over some leaf litter. So it's a little more challenging to get the water. And we found that eDNA has a tremendously higher detection probability. With just a couple bottles of water, we could have nearly a 100% detection probability, something like 98% detection probability. And so we could, in a short amount of time and relatively cheaply, survey for the species in these small headwater streams without being as destructive as the traditional methods mandated. So since then, we've deployed these surveys to more than 150 other streams in Georgia, South Carolina, and in North Carolina. And we've discovered 4 new streams using environmental DNA, and we've later confirmed that a salamander lives there the old-fashioned way. And so this has been really great in expanding the known distribution of the species and figuring out other places to look. We've got some more surveys planned in the near future in Georgia. So the second case study brings about a very different salamander and a very different habitat. And so instead of that tiny trickle of a headwater stream where the Patchnosed salamander lives, here's a picture of Tennessee's Hiwassee River. This is a much larger stream with some much larger salamanders that live in it, including the hellbender. That's not the focus of today's presentation, though. I want to talk to you about Waterdogs in the genus Necturus. These are not plethodonid or lungless salamanders. These are in a different family. And so it was long thought that there was a single species in this genus that lived in the area. That's the mud puppy, Necturus maculosus. This species is primarily found in the Great Lakes region, but it's also known from some large Appalachian streams, including in Georgia. And not too long ago, some of my colleagues at University of Tennessee, Knoxville, realized that this really small stretch of the Hiwassee River, only a couple kilometers, there was not just one species, the mud puppy, but there was a second species of water dog that was sympatric and lived in the exact same reaches. This suffers from that second problem I told you about of being morphologically cryptic, and that they tell me they can tell them apart, but I swear to you, I cannot, and I can identify salamanders all right in general. And so surveying these streams, we wanted to better understand where the salamander lives, and it was often hard to find them, and then, at least for me, difficult to identify them too. So we thought maybe we'd use environmental DNA here too. Instead of developing a separate species-specific assay for each of these water dogs, we decided to use a metabarcoding assay and then look at sequence differences to figure out which water dogs were present. This is a little different than most applications of metabarcoding, including the one I'll tell you about in a minute, in that we're targeting a very small number of species, just 2 in this case. And we're hopefully trying to exclude DNA from all the other aquatic wildlife in the stream. There were some real challenges in the system. One, there were limited genetic resources. We only caught maybe a couple dozen of these salamanders ever. And we only amplified a few genetic loci. So just designing the assay was a little bit challenging. The system is also tough in that in the stream where they're known from, the Hiwassee River, but also neighboring streams that we're interested in surveying, they have a much higher water volume than systems we've traditionally worked in with salamanders. And we weren't sure if the density of the water dogs would be high enough. And in the Hiwassee and again, other rivers in the area, they are all dammed and they're really dynamic hydrologically in that the flow can vary tremendously just in a couple hours span. And I think collectively, we still don't really understand exactly how this affects the distribution of eDNA. So we've had some challenges with this. It doesn't work perfect, but we've used this method with some success to survey a bunch of other large streams in the area. And what you see in orange here is that we found the common species, the mud puppy, in just about everywhere we've looked, including some places where we didn't know it lived before. But we've only found this new species that's still unnamed, this new water dog, in the original place where it was found. It's shown there with a red dot, and just downstream of there, which is shown in the black dot. So it seems that this species might really live just in a tiny area, which doesn't make a whole lot of biogeographic sense, but that's a story for another presentation. But this eDNA has been useful here in sort of confirming what we thought we knew about its very limited distribution. Finally, I would like to talk to you about this third application where we're most excited about expanding these methods. And that's perhaps you don't want to survey for just one species of salamander, like my first example, or two species, like the second. Maybe you want to survey these really diverse Appalachian salamander communities that I hinted at the beginning of the presentation. In just a single stream in some parts of the Southern Appalachia, you can find more than a dozen species. Again, and some of them are behaviorally cryptic. Many of them are morphologically difficult to distinguish. And so this is ripe for a metabarcoding assay. And some years ago, we developed one of these using a mitochondrial marker. And we optimized this for these plethodonid salamanders. And we use it as sort of a case study in this quadruple indexing Illumina Amplicon library method that Travis Glenn's lab developed. And this is a method with much broader appeal for all kinds of projects, from microbiomes to things like this. But we use this as one example to test that method. And so we collect samples the same way, but we amplify all salamander DNA, or probably all amphibian DNA that's in there, and sequence it. And we use differences in the sequences to figure out everything that's there, and something maybe about their relative abundances. And so I'm showing you here just a figure from one of our pilot studies. We collected 3 water samples each from 3 sister tributaries at Coweta LTER, just across the border from Georgia. And we did some technical replicates of each sample. And each color here shows you the number of sequences we got back from each species. So each color is a different plethodonid salamander species. And the details aren't as important here as the general story, which is that we detected all the species we expected to, including some very large, fully aquatic species and some very small species that don't actually have an aquatic life stage, but live in the margins of the stream and still deposit some DNA into there, apparently. You can also see a lot of variation between streams and between samples and between technical replicates of samples. And so this is the sort of variation that's really interesting to understand, and we hope to better explore in the future. But it does seem like this method works pretty well at giving us a first pass, at least, at presence absence of all the salamanders that are there. I just wanted to tell you really quick about one application of this method that we've used. So we partnered with our friends at Great Smoky Mountains Institute at Tremont. It's an educational institute located within the boundaries of Great Smoky Mountains National Park. And for more than 15 years, citizen science volunteers at Tremont have been putting out those leaf litter bags to survey and monitor plethodonid salamander communities and streams around Tremont. And for a year, we had the citizen science volunteers collect eDNA samples paired with their traditional leaf litter bag surveys. Here's a picture of some of these citizen science volunteers collecting a bottle of eDNA. We learned a couple things through this. One is that it seems in our early indications, maybe we can pick up some cool seasonal fluctuations in the eDNA that maybe reflect salamander reproductive seasons. We also learned that, we underscore the difficulty in identifying these salamanders. One species that the citizen scientists had thought was very common in the streams maybe doesn't actually even live there, or is super rare. And this is no fault of the citizen scientists. This is a challenge that even plethodonid salamander biologists have. It's really darn hard to figure out exactly which species are where. This is one value of the eDNA, is that you have a relatively objective taxonomic standard if you have good sequences for the different species. We also learned that if you collect a bottle of water and you leave it behind you for a few minutes as you go walk to collect your next one, a bear might discover that shiny object and chew on it and decrease your sample size a little bit. But this has been a fun project, and it's a good example of how we hope to use these methods in the future for a variety of purposes. So I thought I'd just highlight really quick some things that we're excited about, we'd like to work on in the future. And if any of y'all have good ideas about these, we're all ears. We have a lot to learn about how to apply these methods. So we'd love to sort of adapt them for nanopore sequencing that maybe we can do in real time, even at a field site, to better understand what salamanders are there or other aquatic organisms. The real thing that we all want to know with eDNA is how does it vary spatially and temporally, and how do we interpret those sequence reads to tell us something about the real abundances of the critters in the environment? That's challenging, and people have made great progress in other systems, and we have a lot to learn with these salamanders. And then finally, my lab largely studies contact zones and hybridization and reticulate evolution in salamanders. And it's for all the reasons I told you before, it's sometimes challenging to identify where exactly these different species come in contact and maybe hybridize. And if we can use eDNA to better figure that out, or perhaps more efficiently, that could be a great way to pair these sort of different parts of our lab's research program together. So with that, I'd just like to thank y'all for having me here today. I hope you found it interesting. I'd like to quickly thank our various sources of funding through the years. And I'm excited to talk to y'all in the question and answer session. And I'll see you soon there.